The UNITE Database 37 was launched in 2003 and is a reference database for the molecular identification of fungal, and since 2018 all eukaryotic, species with the nuclear ribosomal internal transcribed spacer, its genetic marker region. This database is based on the concept of species hypotheses. You choose the percent at which you want to work, and the sequences are sorted in comparison to sequences obtained from voucher specimens identified by experts. Diet, barcode. Diet, barcode 38 database was first published under the name R-Assist. Diatom 39 in 2016 starting with data from two sources, the Thon and Culture Collection TCC in the Hydrobiological Station of the French National Institute for Agricultural Research INRA and from the NCBI National Center for Biotechnology Information Nucleotide Database. Diet, barcode provides data for two genetic markers, RBCL ribulose 1, 5-bisphosphate carboxylase oxygenase and 18S ribosomal RNA. The database also involves additional trait information of species, like morphological characteristics, biovolume, size dimensions, etc., lifeforms mobility, colony type, etc. or ecological features, pollution sensitivity, etc. Bioinformatic analysis. In order to obtain well-structured, clean and interpretable data, raw sequencing data must be processed using bioinformatic analysis. The FASTQ file with the sequencing data contains two types of information, the sequences detected in the sample FASTA file and a quality file with quality scores Phred scores associated with each nucleotide of each DNA sequence. The Phred scores indicate the probability with which the associated nucleotide has been correctly scored. Phred quality score and the associated certainty level. In general, the Phred score decreases towards the end of each DNA sequence. Thus some bioinformatics pipelines simply cut the end of the sequences at a defined threshold. Some sequencing technologies, like MIS-seq, use paired-end sequencing during which sequencing is performed from both directions producing better quality. The overlapping sequences are then aligned into contigs and merged. Usually, several samples are pooled in one run, and each sample is characterized by a short DNA fragment, the tag. In a demultiplexing step, sequences are sorted using these tags to reassemble the separate samples. Before further analysis, tags and other adapters are removed from the barcoding sequence DNA fragment. During trimming, the bad quality sequences, low Phred scores, or sequences that are much shorter or longer than the targeted DNA barcode, are removed. The following dereplication step is the process where all of the quality filtered sequences are collapsed into a set of unique reads, individual sequence units ESUs, with the information of their abundance in the samples. After that, chimeras, i.e. compound sequences formed from pieces of mixed origin, are detected and removed. Finally, the sequences are clustered into OTIS, operational taxonomic units, using one of many clustering strategies. The most frequently used bioinformatics software include Mothr, 40 Aparse, 41 Keen, 42 Galaxy, 43 Obitools, 44 JAMP, 45 Bark, 46 and Dada2. 47. Comparing the abundance of reads, i.e. sequences, between different samples is still a challenge because both the total number of reads in a sample as well as the relative amount of reads for a species can vary between samples, methods, or other variables. For comparison, one may then reduce the number of reads of each sample to the minimal number of reads of the samples to be compared, a process called rarefaction. Another way is to use the relative abundance of reads. 48. Species identification and taxonomic assignment. The taxonomic assignment of the OTIS to species is achieved by matching of sequences to reference libraries. The basic local alignment search tool, BLAST, is commonly used to identify regions of similarity between sequences by comparing sequence reads from a sample to sequences in reference databases. 49. If the reference database contains sequences of the relevant species, then the sample sequences can be identified to species level. If a sequence cannot be matched to an existing reference library entry, DNA barcoding can be used to create a new entry. In some cases, due to the incompleteness of reference databases, identification can only be achieved at higher taxonomic levels, such as assignment to a family or class. In some organism groups such as bacteria, taxonomic assignment to species level is often not possible. In such cases, a sample may be assigned to a particular operational taxonomic unit, OTU. In some cases, specimens with identical COI DNA barcodes clearly belong to different species, e.g. species of the fish genus Chromis. 50. Applications. Applications of DNA barcoding include identification of new species, safety assessment of food, identification and assessment of cryptic species, detection of alien species, identification of endangered and threatened species, 51 linking egg and larval stages to adult species, securing intellectual property rights for bioresources, framing global management plans for conservation strategies, elucidate feeding niches, 52 and forensic science. 53 DNA barcode markers can be applied to address basic questions in systematics, ecology, evolutionary biology and conservation, including community assembly, species interaction networks, taxonomic discovery, and assessing priority areas for environmental protection. Identification of species. Specific short DNA sequences or markers from a standardized region of the genome can provide a DNA barcode for identifying species. 54 molecular methods are especially useful when traditional methods are not applicable. DNA barcoding has great applicability in identification of larvae for which there are generally few diagnostic characters available, and in association of different life stages e.g. larval and adult in many animals. 55 Identification of species listed in the Convention of the International Trade of Endangered Species, CITES, appendixes using barcoding techniques is used in monitoring of illegal trade. 56 Detection of invasive species. Alien species can be detected via barcoding. 57-58 Barcoding can be suitable for detection of species in e.g. border control, where rapid and accurate morphological identification is often not possible due to similarities between different species, lack of sufficient diagnostic characteristics 57 and/or lack of taxonomic expertise. Barcoding and metabarcoding can also be used to screen ecosystems for invasive species, and to distinguish between an invasive species and native, morphologically similar, species. 59 The high efficiency of DNA identification is shown relative to the traditional monitoring of biological invasions. 60. Delimiting cryptic species. DNA barcoding enables the identification and recognition of cryptic species. 61 The results of DNA barcoding analyses depend however upon the choice of analytical methods, so the process of delimiting cryptic species using DNA barcodes can be as subjective as any other form of taxonomy. Hebert et al. 2004 concluded that the butterfly Astraps fulgurator in northwestern Costa Rica actually consists of 10 different species. 62 These results, however, were subsequently challenged by Brouwer 2006 who pointed out numerous serious flaws in the analysis, and concluded that the original data could support no more than the possibility of 3 to 7 cryptic taxa rather than 10 cryptic species. 63 Smith et al. 2007 used cytochrome C oxidase I DNA barcodes for species identification of the 20 morphospecies of Belvosia parasitoid flies Diptera, tachinidea reared from caterpillars Lepidoptera in Area de Conservation Guanacaste ACG, northwestern Costa Rica. These authors discovered that barcoding raises the species count to 32, by revealing that each of the 3 parasitoid species, previously considered as generalists, actually are arrays of highly host-specific cryptic species. 64 For 15 morphospecies of polychaetes within the deep Antarctic benthos studied through DNA barcoding, cryptic diversity was found in 50% of the cases. Furthermore, 10 previously overlooked morphospecies were detected, increasing the total species richness in the sample by 233%. 65. Barcoding is a tool to vouch for food quality. Here, DNA from traditional Norwegian Christmas food is extracted at the Molecular Systematic Lab at NTNU University Museum. Diet analysis and food web application. DNA barcoding and metabarcoding can be useful in diet analysis studies, 66, and is typically used if prey specimens cannot be identified based on morphological characters. 67-68 There is a range of sampling approaches in diet analysis. DNA metabarcoding can be conducted on stomach contents, 69 feces, 68-70 saliva, 71, or whole body analysis. 51-72 In fecal samples or highly digested stomach contents, it is often not possible to distinguish tissue from single species, and therefore metabarcoding can be applied instead. 68-73 Feces or saliva represent non-invasive sampling approaches, while whole body analysis often means that the individual needs to be killed first. For smaller organisms, sequencing for stomach content is then often done by sequencing the entire animal. Barcoding for food safety. DNA barcoding represents an essential tool to evaluate the quality of food products. The purpose is to guarantee food traceability, to minimize food piracy, and to evaluate local and typical agro-food production. Another purpose is to safeguard public health. For example, metabarcoding offers the possibility to identify groupers causing ciguatera fish poisoning from meal remnants, 74, or to separate poisonous mushrooms from edible ones, ref. Biomonitoring and ecological assessment. DNA barcoding can be used to assess the presence of endangered species for conservation efforts, ref, or the presence of indicator species reflective to specific ecological conditions, ref, for example excess nutrients or low oxygen levels. Forensic science. DNA barcoding is often used for species identification in forensic science cases. Unknown animal or plant samples at crime scenes can be found, collected, and identified, in hopes of linking it to a suspect and getting a conviction. 75 Poaching, killing of endangered species, and animal abuse are examples of crimes where DNA barcoding is used, since animal DNA is often found. 53-76 On the other hand, plant DNA is usually used as trace evidence to link a suspect to a crime scene. 77. Potentials and shortcomings. Potentials. Traditional bioassessment methods are well established internationally, and serve biomonitoring well, as for example for aquatic bioassessment within the EU directives WFD and MSFD. However, DNA barcoding could improve traditional methods for the following reasons. DNA barcoding can increase taxonomic resolution and harmonize the identification of taxa which are difficult to identify or lack experts. Can more accurately precisely relate environmental factors to specific taxa. Can increase comparability among regions. IV allows for the inclusion of early life stages and fragmented specimens. V allows delimitation of cryptic, rare species. V allows for development of new indices e.g. rare, cryptic species which may be sensitive, tolerant to stressors. V increases the number of samples which can be processed and reduces processing time resulting in increased knowledge of species ecology. V is a non-invasive way of monitoring when using EDNA methods. 78. Time and cost. DNA barcoding is faster than traditional morphological methods all the way from training through to taxonomic assignment. It takes less time to gain expertise in DNA methods than becoming an expert in taxonomy. In addition, the DNA barcoding workflow, i.e. from sample to result, is generally quicker than traditional morphological workflow and allows the processing of more samples. Taxonomic resolution. DNA barcoding allows the resolution of taxa from higher, e.g. family, to lower, e.g. species, taxonomic levels, that are otherwise too difficult to identify using traditional morphological methods, like e.g. identification via microscopy. For example, Chironomidae, the non-biting midge, are widely distributed in both terrestrial and freshwater ecosystems. Their richness and abundance make them important for ecological processes and networks, and they are one of many invertebrate groups used in biomonitoring. Invertebrate samples can contain as many as 100 species of chironomids, which often make up as much as 50% of a sample. Despite this, they are usually not identified below the family level because of the taxonomic expertise and time required. 79. This may result in different chironomid species with different ecological preferences grouped together, resulting in inaccurate assessment of water quality. DNA barcoding provides the opportunity to resolve taxa, and directly relates stressor effects to specific taxa such as individual chironomid species. For example, Behrman et al. 2018 DNA barcoded chironomidae to investigate their response to multiple stressors, reduced flow, increased fine sediment and increased salinity. 80. After barcoding, it was found that the chironomid sample consisted of 183 operational taxonomic units, OTIS, i.e. barcodes, sequences, that are often equivalent to morphological species. These 183 OTIS displayed 15 response types rather than the previously reported 81 2 response types recorded when all chironomids were grouped together in the same multiple stressor study. A similar trend was discovered in a study by Mocker et al. 2016, which discovered cryptic diversity within the New Zealand mayfly species Deleitidium sp. w.terrain.net.nz/friends-of-te-group/invertebrates-freshwater-new-Zealand/mayfly-nymph-genus Deleitidium, HTML. This study found different response patterns of 12 molecular distinct OTIS to stressors, which may change the consensus that this mayfly is sensitive to pollution. 82. Shortcomings. Despite the advantages offered by DNA barcoding, it has also been suggested that DNA barcoding is best used as a complement to traditional morphological methods. 78. This recommendation is based on multiple perceived challenges. Physical parameters. It is not completely straightforward to connect DNA barcodes with ecological preferences of the barcoded taxon in question, as is needed if barcoding is to be used for biomonitoring. For example, detecting target DNA in aquatic systems depends on the concentration of DNA molecules at a site, which in turn can be affected by many factors. The presence of DNA molecules also depends on dispersion at a site, e.g. direction or strength of currents. It is not really known how DNA moves around in streams and lakes, which makes sampling difficult. Another factor might be the behavior of the target species, e.g. fish can have seasonal changes of movements, crayfish or mussels will release DNA in larger amounts just at certain times of their life, molting, spawning. For DNA in soil, even less is known about distribution, quantity or quality. The major limitation of the barcoding method is that it relies on barcode reference libraries for the taxonomic identification of the sequences. The taxonomic identification is accurate only if a reliable reference is available. However, most databases are still incomplete, especially for smaller organisms e.g. fungi, phytoplankton, nematoda etc. In addition, current databases contain misidentifications, spelling mistakes and other errors. There is massive curation and completion effort around the databases for all organisms necessary, involving large barcoding projects, for example the IBOL project for the Barcode of Life Data Systems Bold reference database. 83-84. However, completion and curation are difficult and time-consuming. Without vouchered specimens, there can be no certainty about whether the sequence used as a reference is correct. DNA sequence databases like GenBank contain many sequences that are not tied to vouchered specimens, for example, herbarium specimens, cultured cell lines, or sometimes images. This is problematic in the face of taxonomic issues such as whether several species should be split or combined, or whether past identifications were sound. Reusing sequences, not tied to vouchered specimens, of initially misidentified organism may support incorrect conclusions and must be avoided. 85. Therefore, best practice for DNA barcoding is to sequence vouchered specimens. 86-87. For many taxa, it can be however difficult to obtain reference specimens, for example with specimens that are difficult to catch, available specimens are poorly conserved, or adequate taxonomic expertise is lacking. 85. Importantly, DNA barcodes can also be used to create interim taxonomy, in which case OTIS can be used as substitutes for traditional Latin binomials, thus significantly reducing dependency on fully populated reference databases. 88. Technological bias. DNA barcoding also carries methodological bias, from sampling to bioinformatics data analysis. Beside the risk of contamination of the DNA sample by PCR inhibitors, primer bias is one of the major sources of errors in DNA barcoding. 89-90. The isolation of an efficient DNA marker and the design of primers is a complex process, and considerable effort has been made to develop primers for DNA barcoding in different taxonomic groups. 91. However, primers will often bind preferentially to some sequences, leading to differential primer efficiency and specificity in unrepresentative communities, assessment and richness inflation. 92. Thus, the composition of the sample's community's sequences is mainly altered at the PCR step. Besides, PCR replication is often required, but leads to an exponential increase in the risk of contamination. Several studies have highlighted the possibility to use mitochondria-enriched samples, 93-94, or PCR-free approaches to avoid these biases, but as of 2018, the DNA metabarcoding technique is still based on the sequencing of amplicons. 91. Other biases enter the picture during the sequencing and during the bioinformatic processing of the sequences, like the creation of chimeras. Lack of standardization. Even as DNA barcoding is more widely used and applied, there is no agreement concerning the methods for DNA preservation or extraction, the choices of DNA markers and primers set, or PCR protocols. The parameters of bioinformatics pipelines, for example OTU clustering, taxonomic assignment algorithms or thresholds etc., are at the origin of much debate among DNA barcoding users. 91. Sequencing technologies are also rapidly evolving, together with the tools for the analysis of the massive amounts of DNA data generated, and standardization of the methods is urgently needed to enable collaboration and data sharing at greater spatial and time scale. This standardization of barcoding methods at the European scale is part of the objectives of the European Cost Action in Aquanet, 95, and is also addressed by CEN, the European Committee for Standardization. 96. Another criticism of DNA barcoding is its limited efficiency for accurate discrimination below species level, for example, to distinguish between varieties for hybrid detection, and that it can be affected by evolutionary rates. Mismatches between conventional, morphological, and barcode-based identification. It is important to know that taxa lists derived by conventional, morphological identification are not, and maybe never will be, directly comparable to taxa lists derived from barcode-based identification because of several reasons. The most important cause is probably the incompleteness and lack of accuracy of the molecular reference databases preventing a correct taxonomic assignment of EDNA sequences. Taxa not present in reference databases will not be found by EDNA, and sequences linked to a wrong name will lead to incorrect identification. 78. Other known causes are a different sampling scale and size between a traditional and a molecular sample, the possible analysis of dead organisms, which can happen in different ways for both methods depending on organism group, and the specific selection of identification in either method, i.e. varying taxonomical expertise or possibility to identify certain organism groups, respectively primer bias leading also to a potential biased analysis of taxa. 78. Estimates of richness, diversity. DNA barcoding can result in an over- or underestimate of species richness and diversity. Some studies suggest that artifacts, identification of species not present in a community, are a major cause of inflated biodiversity. 97-98. The most problematic issue are taxa represented by low numbers of sequencing reads. These reads are usually removed during the data filtering process, since different studies suggest that most of these low-frequency reads may be artifacts. 99. However, real rare taxa may exist among these low-abundance reads. 100. Rare sequences can reflect unique lineages in communities which make them informative and valuable sequences. Thus, there is a strong need for more robust bioinformatics algorithms that allow the differentiation between informative reads and artifacts. Complete reference libraries would also allow a better testing of bioinformatics algorithms, by permitting a better filtering of artifacts, i.e. the removal of sequences lacking a counterpart among extant species, and therefore, it would be possible to obtain a more accurate species assignment. 101. Cryptic diversity can also result in inflated biodiversity as one morphological species may actually split into many distinct molecular sequences. 78. This will go a long way in generating DNA reference data, which is crucial for environmental DNA-based biodiversity monitoring. Megabarcoding. Megabarcoding is a term used to describe high-throughput specimen-based DNA barcoding, where thousands of specimens can be barcoded simultaneously for species identification and discovery. 102, 103, 104, 105, 106. Megabarcoding workflow. This is enabled by the use of third-generation sequencing platforms including PacBio, SQLite2 by Pacific Biosciences and Minion, ProMethION by Oxford Nanopore Technology. As compared to Sanger sequencing, megabarcoding is faster and cheaper, allowing for the large-scale generation of DNA barcodes for thousands of species. 107. Applications. Megabarcoding can help fill the dark taxa. DNA barcode reference data gap for insects and accelerates species discovery. 108, 109 understands species diversity patterns. 110, 111, 112 evaluate species richness. 113 generate rapid biodiversity species inventories. 114 track baseline shifts. 115 and matching life history stages. 116. Metabarcoding. Differences in the standard methods for DNA barcoding and metabarcoding. While DNA barcoding points to find a specific species, metabarcoding looks for the whole community. Metabarcoding is defined as the barcoding of DNA or EDNA, environmental DNA, that allows for simultaneous identification of many taxa within the same environmental sample, however often within the same organism group. The main difference between the approaches is that metabarcoding, in contrast to barcoding, does not focus on one specific organism, but instead aims to determine species composition within a sample. Methodology. The metabarcoding procedure, like general barcoding, covers the steps of DNA extraction, PCR amplification, sequencing and data analysis. A barcode consists of a short variable gene region, for example, see different markers barcodes, which is useful for taxonomic assignment flanked by highly conserved gene regions which can be used for primer design. 117 different genes are used depending if the aim is to barcode single species or metabarcoding several species. In the latter case, a more universal gene is used. Metabarcoding does not use single species DNA, RNA as a starting point, but DNA, RNA from several different organisms derived from one environmental or bulk sample. Applications. Metabarcoding has the potential to complement biodiversity measures, and even replace them in some instances, especially as the technology advances and procedures gradually become cheaper, more optimized and widespread. 118, 119. DNA metabarcoding applications include biodiversity monitoring in terrestrial and aquatic environments, paleontology and ancient ecosystems, plant-pollinator interactions, diet analysis and food safety. Advantages and challenges. The general advantages and shortcomings for barcoding reviewed above are valid also for metabarcoding. One particular drawback for metabarcoding studies is that there is no consensus yet regarding the optimal experimental 97-98 The most problematic issue are taxa represented by low numbers of sequencing reads. These reads are usually removed during the data filtering process, since different studies suggest that most of these low-frequency reads may be artifacts. 99 However, real rare taxa may exist among these low-abundance reads. 100 rare sequences can reflect unique lineages in communities, which make them informative and valuable sequences. Thus, there is a strong need for more robust bioinformatics algorithms that allow the differentiation between informative reads and artifacts. Complete reference libraries would also allow a better testing of bioinformatics algorithms, by permitting a better filtering of artifacts, i.e. the removal of sequences lacking a counterpart among extant species, and therefore, it would be possible to obtain a more accurate species assignment. 101 Cryptic diversity can also result in inflated biodiversity, as one morphological species may actually split into many distinct molecular sequences. 78 This will go a long way in generating DNA reference data, which is crucial for environmental DNA-based biodiversity monitoring. Megabarcoding. Megabarcoding is a term used to describe high-throughput specimen-based DNA barcoding, where thousands of specimens can be barcoded simultaneously for species identification and discovery. 102, 103, 104, 105, 106. Megabarcoding workflow. This is enabled by the use of third-generation sequencing platforms including PacBio, SQLite2 by Pacific Biosciences and Minion, ProMethION by Oxford Nanopore Technology. As compared to Sanger sequencing, megabarcoding is faster and cheaper, allowing for the large-scale generation of DNA barcodes for thousands of species. 107. Applications. Megabarcoding can help fill the dark taxa. DNA barcode reference data gap for insects and accelerate species discovery. 108, 109 Understand species diversity patterns. 110, 111, 112 Evaluate species richness. 113 Generate rapid biodiversity species inventories. 114 Track baseline shifts. 115 And matching life history stages. 116. Metabarcoding. Differences in the standard methods for DNA barcoding and metabarcoding. While DNA barcoding points to find a specific species, metabarcoding looks for the whole community. Metabarcoding is defined as the barcoding of DNA or EDNA, environmental DNA, that allows for simultaneous identification of many taxa within the same environmental sample, however often within the same organism group. The main difference between the approaches is that metabarcoding, in contrast to barcoding, does not focus on one specific organism, but instead aims to determine species composition within a sample. Methodology. The metabarcoding procedure, like general barcoding, covers the steps of DNA extraction, PCR amplification, sequencing and data analysis. A barcode consists of a short variable gene region, for example, see different markers barcodes, which is useful for taxonomic assignment flanked by highly conserved gene regions which can be used for primer design. 117 Different genes are used depending if the aim is to barcode single species or metabarcoding several species. In the latter case, a more universal gene is used. Metabarcoding does not use single species DNA, RNA as a starting point, but DNA, RNA from several different organisms derived from one environmental or bulk sample. Applications. Metabarcoding has the potential to complement biodiversity measures, and even replace them in some instances, especially as the technology advances and procedures gradually become cheaper, more optimized and widespread. 118, 119. DNA metabarcoding applications include biodiversity monitoring in terrestrial and aquatic environments, paleontology and ancient ecosystems, plant-pollinator interactions, diet analysis and food safety. Advantages and challenges. The general advantages and shortcomings for barcoding reviewed above are valid also for metabarcoding. One particular drawback for metabarcoding studies is that there is no consensus yet regarding the optimal experimental design and bioinformatics criteria to be applied in EDNA metabarcoding. 120 However, there are current joint attempts, like e.g. the EU Cost Network DNAQUANet, to move forward by exchanging experience and knowledge to establish best practice standards for biomonitoring. 78. Artificial DNA barcoding. In 2014, researchers from ETH Zurich suggested using artificial, sub-micrometer-sized DNA barcodes as an invisible oil tag. The barcodes consist of synthetic DNA sequences inside magnetically recoverable silica particles. They can be added to food oil in a very small amount, down to 1 ppb, as a label, and can be retrieved at any time for authenticity test by PCR sequencing. This method can be used to test olive oil for adulteration. In 2014, researchers from ETH Zurich suggested using artificial, sub-micrometer-sized DNA barcodes as an invisible oil tag. The barcodes consist of synthetic DNA sequences inside magnetically recoverable silica particles. They can be added to food oil in a very small amount, down to 1 ppb, as a label, and can be retrieved at any time for authenticity test by PCR sequencing. This method can be used to test olive oil for adulteration. Disease ecology. Disease ecology is a subdiscipline of ecology concerned with the mechanisms, patterns, and effects of host-pathogen interactions, particularly those of infectious diseases. For example, it examines how parasites spread through and influence wildlife populations and communities. 2. By studying the flow of diseases within the natural environment, scientists seek to better understand how changes within our environment can shape how pathogens, and other diseases, travel. 2. Therefore, diseases ecology seeks to understand the links between ecological interactions and disease evolution. 2. New emerging and re-emerging infectious diseases, infecting both wildlife and humans, are increasing at unprecedented rates which can have lasting impacts on public health, ecosystem health, and biodiversity. 3. Factors affecting spread of diseases. Parasitic infections, along with certain transmitted diseases, are present in wildlife which can have severe health effects on particular individuals and populations. 4. Constant host-parasite interactions make disease ecology critical in conservation ecology. 4. Ecological factors. Ecological factors that can determine the persistence and the spread of diseases are population size, density, and composition. 4. Host population size is important in the context of host-parasite interactions since the spread of diseases needs a host population large enough to sustain parasitic interactions. The health of the overall population and the size of the weakened population members will also influence the way that parasites and diseases will transmit among members. 4. Additionally, competition and predation dynamics in the ecosystem can influence the density of potential hosts which can either propagate or limit the spread of diseases. 4. Predator-prey interactions. In some cases when a parasite has weakened an animal it will become easier prey for a predator species. 5. Occasionally predators will prefer feeding on the sick or infected prey even though they carry a parasite because of the opportunity weak prey present. 5. Without the presence of a predator species the prey species would likely exceed manageable numbers therefore leading to the rapid spread of pathogens throughout the prey population. 6. Available host numbers increased when the infected individuals are not removed due to low predation. 7. However, there are some situations where predator feeding can disturb a pathogen that previously was dormant leading to an epidemic that otherwise would not have occurred. 8. Some parasites are able to survive when their host species is consumed leading to the parasite being distributed in the waste of the predator which can continue the spread of disease. 9. Parasitism. Parasitism in disease ecology is important because it can shape the way many habitats function because they are disease carriers. These diseases can alter the timing of events, biogeochemical cycles, and even the flow of energy in a habitat. 10. Parasites are able to limit population growth and reproduction of species which may lead to a shift in the balance of an ecosystem. 11. Other ways parasites impact systems are through nutrient cycles. Parasites are able to create imbalances of the elements in a system through the relationship they have with a host and the host's diet. 12. Biological factors. Biological factors that can determine the persistence of diseases include parameters pertaining at the level of the individual within the population, one single organism. 4. Sex differences are found to be prevalent in disease transmission. For example, male American minks are larger and travel wider distances, making them more prone to come into contact with parasites and diseases. The host species age may additionally affect the rate in which diseases are transmitted. Younger members of populations have yet to acquire herd immunity and are therefore more susceptible to parasitic infections. 4. Anthropogenic factors. Anthropogenic factors of disease spread can be through the introduction or translocation of wildlife for conservation purposes by humans. 4. Additionally, human activity is changing the way in which diseases move through the natural environment. 4. In relation to anthropogenic factors. Humans are strongly impacting how disease is spread by creating what is known as novel species associations. 13. Globalization, mainly through world travel and trade, has created a system in which pathogens and other species are more in contact with one another than before. 13, 14, 15. Ecological disruption, including habitat fragmentation and road construction, degrade natural landscapes and have been studied as drivers of recent emergence and re-emergence of infectious diseases worldwide. 14. Scientists have speculated that habitat destruction and biodiversity loss are some of the main reasons influencing the rapid spread of non-human, disease-carrying vectors. The loss of predators, that mitigate the ability for pathogen transmission, can increase the rate of disease transmission. 14. Human anthropogenic-induced climate change is becoming problematic, as parasites and their associated diseases can move to higher latitudes with increasing global temperatures. New diseases can therefore infect populations that were previously never in contact with certain pathogens. 13. Urbanization and biodiversity loss. Urban sprawl of Toronto, Canada, viewed from the CN Tower. Urbanization is considered one of the main land use changes, defined as the growth in the area and number of people inhabiting cities and creates artificial landscapes that build up structures for human use. 16. With over 65% of the global human population living in cities by 2025, ecological impacts of urbanization focuses mainly on biodiversity loss defined as the decline in species richness. 16. With empirical evidence, scientists are understanding that biodiversity loss is associated with increased disease transmission and worsening of disease severity for humans, wildlife, and certain plant species. 17. As biodiversity is lost worldwide, it is oftentimes the larger, slower reproducing animal species that will go extinct first. This leaves smaller, more adaptable, fast reproducing species abundant. 18. Research has shown that these smaller species are more likely the ones to carry and transmit pathogens. Key examples include bats, rats, and mice. 18. Invasive species. Globalization, especially world trade and travel, has facilitated the spread of non-native species worldwide. 15. Newly introduced invasive species have the ability to alter ecological dynamics through local and regional extinction of native species. 19. This can promote changes to the ecosystem including the shift in abundance and richness of native species. 19. New invasive species, and the diseases they potentially carry, can escape into the environment and alter the existing natural ecosystems and the ecosystem services that people are dependent upon, including water quality and nutrient availability. 19. Habitat fragmentation. Highways can cause habitat fragmentation which increases edge effects and promotes disease spread. Encroachment on natural ecosystems and wildlife with rapid urbanization exposes humans to a wide variety of disease-carrying animals. 20. Habitat fragmentation leads to increased edge effects and increases the contact between different communities, vectors, and pathogens which can increase disease transmission. 21. It is argued that between 2013 and 2015, the Ebola virus disease (EDB) outbreak in West Africa began due to deforestation and habitat degradation. 22. In this case, frugivorous and insectivorous bat species had less forest serving as a barrier between them and dense human settlements. 22. Transmission of the Ebola virus is believed to have occurred through direct contact with bat species carrying the pathogen and humans, encroaching on natural ecosystems. 22. Climate change. Scientists have deemed vector-borne diseases to be sensitive to changes in weather and climate. 23. The abundance of disease-carrying vectors in the environment depends on multiple factors, including temperature, relative humidity, and water availability, all factors necessary for the reproductive processes and success of disease-carrying vectors. 23. Climate change predictions include rising temperatures and changes in rainfall pattern which can create suitable habitats and increases the overall survival rate and fitness of pathogen-carrying species. 24. With a warming climate, pathogens and parasites can begin shifting their native geographic ranges to higher latitudes and infect host species in which they have no prior interaction with. 25. The shift in rainfall patterns can additionally indicate the presence of disease-carrying vectors. 24. For example, mosquitoes spread diseases such as malaria and lymphatic filariasis. 24. The distribution of lymphatic filariasis via mosquitoes can be determined by looking at soil moisture content, an indicator of viable mosquito breeding habitat, as mosquito larvae need shallow, stagnant water to survive. As temperature and precipitation patterns change, so will soil moisture levels in the corresponding mosquito populations. 24. As climate change continues to disrupt ecosystems around the world, it can make both human and non-human populations more or less vulnerable to disease depending on the specific effects of climate change on the disease. 26. The subject of climate change and its impact on disease is increasingly attracting the attention of health professionals and climate change scientists, particularly with respect to malaria and other vector-transmitted human diseases. 27. 1. More specifically, climate change can impact malaria transmissions by extending the season of transmission and creating more breeding sites due to increasing temperatures and rainfall, respectively. 28. Increases in malaria transmissions and other vector-transmitted human diseases can have a devastating impact on communities that do not receive appropriate medical care and on people who have not had exposure to these diseases. 28. 29. In relation to tropical, northern temperate zones, and the Arctic, it is thought that the effects of climate change on temperature will increase with latitude. 30. This means that northern temperate zones will experience more temperature changes than tropical zones. 30. Tropical zones experience less climate variability, so organisms in tropical zones have adjusted to a continuous climate. 30. Therefore, slight disruptions in climate can dramatically affect the organisms in tropical zones. 30. Climate change can affect organisms by elongating their reproductive cycles. 26. In addition to this, climate change allows for pathogens to expand beyond tropical zones, dramatically impacting species because of the introduction of new pathogens. 29. These impacted species include humans and human livestock. 29. Changes in northern temperate zones and the Arctic are also expected. 30. More specifically, the effects of climate change on temperature increase with latitude, so the temperature in northern temperate zones is projected to increase and the temperature in the Arctic is projected to increase even more. 30. Like tropical zones, climate change in northern temperate zones and the Arctic can also cause species to move beyond their original niche. 29. For example, climate change has allowed elk to move north in areas that overlap with other species such as caribou. 29. When the elk move, they introduce new pathogens into the area, thus harming the caribou. 29. Models and predicting disease ecology. There are numerous approaches when predicting the impacts of climate change on diseases. 29. Static approaches use reproduction rates to find how climate change will affect vectors. 29. An example of the use of static approaches is a process-based model called miasma. 26. This model explores the relationship between different climate change scenarios and the reproduction rate of vectors. 26. This model has been used specifically to look at mosquitoes in African highlands to make predictions about the future of the development and feeding of mosquitoes. 31. Additionally, this model can be used to find the population of mosquitoes that bite, allowing predictions of diseases such as dengue fever. 26. Another approach includes statistical-based models, which relies on observations unlike process-based models. 26. An example of this type of model is CLIMEX, which maps vector species over geographical locations while accounting for climate factors. 26. It is important to note that this approach does have limitations. 26. CLIMEX does not include all factors that impact vector species. 26. Time series models can also be used to find how climate change will modify disease dynamics. 29. However, this approach has a downside. Only a limited number of locations and pathogens can be looked at simultaneously using time series models. 29. Predictions of ENSO, El Niño Southern Oscillation, can also help predict diseases. 32. ENSO events can create cooler temperatures in the western tropical Pacific and warmer temperatures in the central and eastern tropical Pacific, leading to intense precipitation and storms. 26. Changes in climate due to ENSO can affect the dynamics of diseases and can affect the water sources humans use. 32. 26. For example, in 1991, cholera reappeared in Peru around the same time as an El Niño event occurred. 32. ENSO events can be anticipated early on, and therefore by predicting ENSO, predictions about disease transmission peaks can be made up to 2 months before they occur. 32. 26. Notable examples in disease ecology. Ticks are a vector for Lyme disease. Barn owls are a host species for West Nile virus. Malaria. Malaria is a disease transferred by the female Anopheles mosquito, located predominantly in sub-Saharan Africa and is a long-withstanding public health issue. 33. It is a disease that is strongly regulated by climate factors and therefore climate change will have a notable impact on the transmission of the disease. 34. 35. As temperatures warm, the reproductive phase of the Plasmodium parasite, within the gut of the female mosquito, will undergo completion. 34. This will ensure that the female mosquito becomes infective before the end of its lifespan. 34. Precipitation is also a critical factor for the breeding and the transmission of malaria, and with climate change influencing regular precipitation patterns, studies are finding that mosquito breeding potential can increase as a direct result of climate change. 35. Lyme disease. Lyme disease is the most common tick-borne disease throughout the United States and Europe with an estimated 476,000 cases in Europe and 200,000 cases in the United States per year. 36. Recently, studies have concluded that there is an increased risk of Lyme disease in southern Canada due to the home range expansion of the tick vector Ixodes scapularis, which is responsible for carrying the disease. 37. Climate change creates milder winters and extended spring and autumn seasons. 38. This creates hospitable habitats for ticks thrive at higher latitudes, where they are normally not found. 38. Human infections of Lyme disease have been increasingly prominent in certain southern parts of Canadian provinces such as Ontario, Quebec, Manitoba, and Nova Scotia. 37. According to Canadian published studies, other environmental factors are contributing to the expansion of the Ixodes scapularis home range, which include the introduction of the vector through migratory birds and density of deer populations. 37. West Nile virus. West Nile virus is transferred between mosquitoes and birds of prey including eagles, hawks, falcons, and owls. 39. In the United States, West Nile virus is being increasingly studied in New York and Connecticut due to the effects of climate change on two disease-carrying vectors. 40. Climate change is promoting the hybridization amongst two mosquito vectors, C. pipiens and C. quinquefasciatus, which can have an effect on the genetic composition of the hybrid, allowing it to become more effective at transmitting diseases and increases its adaptability to different climatic conditions. 40. Epidemiology. Immunology. Microbiology. Natural reservoir. Threshold host density zoonotic diseases Ecoimmunology 1. Ostfeld 2015. 2. Holly and Altizer 2011 pp. 48-60. 3. Plowright et al. 2008 pp. 420-429. 4. Koladzij Sobosinska 2019 pp. 300. Disease. This is the latest. A disease is a particular abnormal condition that adversely affects the structure or function of all or part of an organism and is not immediately due to any external injury. 1. 2. Diseases are often known to be medical conditions that are associated with specific signs and symptoms. A disease may be caused by external factors such as pathogens or by internal dysfunctions. For example, internal dysfunctions of the immune system can produce a variety of different diseases, including various forms of immunodeficiency, hypersensitivity, allergies, and autoimmune disorders. The Sick Girl, by Michael Ancher, 1882, National Gallery of Denmark. In humans, disease is often used more broadly to refer to any condition that causes pain, dysfunction, distress, social problems, or death to the person affected, or similar problems for those in contact with the person. In this broader sense, it sometimes includes injuries, disabilities, disorders, syndromes, infections, isolated symptoms, deviant behaviors, and atypical variations of structure and function, while in other contexts and for other purposes these may be considered distinguishable categories. Diseases can affect people not only physically but also mentally, as contracting and living with a disease can alter the affected person's perspective on life. Death due to disease is called death by natural causes. There are four main types of disease: infectious diseases, deficiency diseases, hereditary diseases including both genetic and non-genetic hereditary diseases, and physiological diseases. Diseases can also be classified in other ways, such as communicable versus non-communicable diseases. The deadliest diseases in humans are coronary artery disease, blood flow obstruction, followed by cerebrovascular disease and lower respiratory infections. 3. In developed countries, the diseases that cause the most sickness overall are neuropsychiatric conditions, such as depression and anxiety. The study of disease is called pathology, which includes the study of etiology, or cause. Terminology. Concepts. In many cases, terms such as disease, disorder, morbidity, sickness and illness are used interchangeably. However, there are situations when specific terms are considered preferable. 4. Disease. The term disease broadly refers to any condition that impairs the normal functioning of the body. For this reason, diseases are associated with the dysfunction of the body's normal homeostatic processes. 5. Commonly, the term is used to refer specifically to infectious diseases, which are clinically evident diseases that result from the presence of pathogenic microbial agents, including viruses, bacteria, fungi, protozoa, multicellular organisms, and aberrant proteins known as prions. An infection or colonization that does not and will not produce clinically evident impairment of normal functioning, such as the presence of the normal bacteria and yeasts in the gut, or of a passenger virus, is not considered a disease. By contrast, an infection that is asymptomatic during its incubation period, but expected to produce symptoms later, is usually considered a disease. Non-infectious diseases are all other diseases, including most forms of cancer, heart disease, and genetic disease. Acquired disease. An acquired disease is one that began at some point during one's lifetime, as opposed to disease that was already present at birth, which is congenital disease. Acquired sounds like it could mean caught via contagion, but it simply means acquired sometime after birth. It also sounds like it could imply secondary disease, but acquired disease can be primary disease. Acute disease. An acute disease is one of a short-term nature. Acute, the term sometimes also connotes a fulminant nature. Chronic condition or chronic disease. A chronic disease is one that persists over time, often for at least 6 months, but may also include illnesses that are expected to last for the entirety of one's natural life. Congenital disorder or congenital disease. A congenital disorder is one that is present at birth. It is often a genetic disease or disorder and can be inherited. It can also be the result of a vertically transmitted infection from the mother, such as HIV/AIDS. Genetic disease. A genetic disorder or disease is caused by one or more genetic mutations. It is often inherited, but some mutations are random and de novo. Hereditary or inherited disease. A hereditary disease is a type of genetic disease caused by genetic mutations that are hereditary and can run in families. Iatrogenic disease. An iatrogenic disease or condition is one that is caused by medical intervention, whether as a side effect of a treatment or as an inadvertent outcome. Idiopathic disease. An idiopathic disease has an unknown cause or source. As medical science has advanced, many diseases with entirely unknown causes have had some aspects of their sources explained and therefore shed their idiopathic status. Iatrogenic disease. An iatrogenic disease or condition is one that is caused by medical intervention, whether as a side effect of a treatment or as an inadvertent outcome. Idiopathic disease. An idiopathic disease has an unknown cause or source. As medical science has advanced, many diseases with entirely unknown causes have had some aspects of their sources explained and therefore shed their idiopathic status. For example, when germs were discovered, it became known that they were a cause of infection, but particular germs and diseases had not been linked. In another example, it is known that autoimmunity is the cause of some forms of diabetes mellitus type 1, even though the particular molecular pathways by which it works are not yet understood. It is also common to know certain factors are associated with certain diseases. However, association does not necessarily imply causality. For example, a third factor might be causing both the disease and the associated phenomenon. Incurable disease. A disease that cannot be cured. Incurable diseases are not necessarily terminal diseases, and sometimes a disease's symptoms can be treated sufficiently for the disease to have little or no impact on quality of life. Primary disease. A primary disease is a disease that is due to a root cause of illness, as opposed to secondary disease, which is a sequela or complication that is caused by the primary disease. For example, a common cold is a primary disease, where rhinitis is a possible secondary disease, or sequela. A doctor must determine what primary disease, a cold or bacterial infection, is causing a patient's secondary rhinitis when deciding whether or not to prescribe antibiotics. Secondary disease. A secondary disease is a disease that is a sequela or complication of a prior, causal disease, which is referred to as the primary disease or simply the underlying cause, root cause. For example, a bacterial infection can be primary, wherein a healthy person is exposed to bacteria and becomes infected, or it can be secondary to a primary cause, that predisposes the body to infection. For example, a primary viral infection that weakens the immune system could lead to a secondary bacterial infection. Similarly, a primary burn that creates an open wound could provide an entry point for bacteria, and led to a secondary bacterial infection. Terminal disease. A terminal disease is one that is expected to have the inevitable result of death. Previously, AIDS was a terminal disease. It is now incurable, but can be managed indefinitely using medications. Illness. The terms illness and sickness are both generally used as synonyms for disease. However, the term illness is occasionally used to refer specifically to the patient's personal experience of their disease. 6, 7, 8, 9. In this model, it is possible for a person to have a disease without being ill, to have an objectively definable, but asymptomatic, medical condition, such as a subclinical infection, or to have a clinically apparent physical impairment but not feel sick or distressed by it, and to be ill without being diseased, such as when a person perceives a normal experience as a medical condition, or medicalizes a non-disease situation in their life. For example, a person who feels unwell as a result of embarrassment, and who interprets those feelings as sickness rather than normal emotions. Symptoms of illness are often not directly the result of infection, but a collection of evolved responses, sickness behavior by the body, that helps clear infection and promote recovery. Such aspects of illness can include lethargy, depression, loss of appetite, sleepiness, hyperalgesia, and inability to concentrate. 10, 11, 12. Disorder. A disorder is a functional abnormality or disturbance that may or may not show specific signs and symptoms. Medical disorders can be categorized into mental disorders, physical disorders, genetic disorders, emotional and behavioral disorders, and functional disorders. 13. The term disorder is often considered more value-neutral and less stigmatizing than the terms disease or illness, and therefore is preferred terminology in some circumstances. 14. In mental health, the term mental disorder is used as a way of acknowledging the complex interaction of biological, social, and psychological factors in psychiatric conditions. However, the term disorder is also used in many other areas of medicine, primarily to identify physical disorders that are not caused by infectious organisms, such as metabolic disorders. Medical condition or health condition. A medical condition or health condition is a broad concept that includes all diseases, lesions, disorders, or non-pathologic condition that normally receives medical treatment, such as pregnancy or childbirth. While the term medical condition generally includes mental illnesses, in some contexts the term is used specifically to denote any illness, injury, or disease except for mental illnesses. 13. The term disorder is often considered more value-neutral and less stigmatizing than the terms disease or illness, and therefore is preferred terminology in some circumstances. 14. In mental health, the term mental disorder is used as a way of acknowledging the complex interaction of biological, social, and psychological factors in psychiatric conditions. However, the term disorder is also used in many other areas of medicine, primarily to identify physical disorders that are not caused by infectious organisms, such as metabolic disorders. Medical disorders can be categorized into mental disorders, physical disorders, genetic disorders, emotional and behavioral disorders, and functional disorders. Medical condition or health condition. A medical condition or health condition is a broad concept that includes all diseases, lesions, disorders, or non-pathologic condition that normally receives medical treatment, such as pregnancy or childbirth. While the term medical condition generally includes mental illnesses, in some contexts the term is used specifically to denote any illness, injury, or disease except for mental illnesses. The Diagnostic and Statistical Manual of Mental Disorders (DSM), the widely used psychiatric manual that defines all mental disorders, uses the term general medical condition to refer to all diseases, illnesses, and injuries except for mental disorders. 15. This usage is also commonly seen in the psychiatric literature. Some health insurance policies also define a medical condition as any illness, injury, or disease except for psychiatric illnesses. 16. As it is more value neutral than terms like disease, the term medical condition is sometimes preferred by people with health issues that they do not consider deleterious. However, by emphasizing the medical nature of the condition, this term is sometimes rejected, such as by proponents of the autism rights movement. The term medical condition is also a synonym for medical state, in which case it describes an individual patient's current state from a medical standpoint. This usage appears in statements that describe a patient as being in critical condition, for example. Morbidity. Morbidity, from Latin morbidus sick, unhealthy, is a diseased state, disability, or poor health due to any cause. 17. The term may refer to the existence of any form of disease, or to the degree that the health condition affects the patient. Among severely ill patients, the level of morbidity is often measured by ICU scoring systems. Comorbidity, or coexisting disease, is the simultaneous presence of two or more medical conditions, such as schizophrenia and substance abuse. In epidemiology and actuarial science, the term morbidity, also morbidity rate or morbidity frequency, can refer to either the incidence rate, the prevalence of a disease or medical condition, or the percentage of people who experience a given condition within a given time frame, e.g., 20% of people will get influenza in a year. 18. This measure of sickness is contrasted with the mortality rate of a condition, which is the proportion of people dying during a given time interval. Morbidity rates are used in actuarial professions, such as health insurance, life insurance, and long-term care insurance, to determine the premiums charged to customers. Morbidity rates help insurers predict the likelihood that an insured will contract or develop any number of specified diseases. Pathosis or pathology. Pathosis, plural pathoses, is synonymous with disease. The word pathology also has this sense. Morbidity, from Latin morbidus sick, unhealthy, is a diseased state, disability, or poor health due to any cause. 17. The term may refer to the existence of any form of disease, or to the degree that the health condition affects the patient. Among severely ill patients, the level of morbidity is often measured by ICU scoring systems. Comorbidity, or coexisting disease, is the simultaneous presence of two or more medical conditions, such as schizophrenia and substance abuse. In epidemiology and actuarial science, the term morbidity, also morbidity rate or morbidity frequency, can refer to either the incidence rate, the prevalence of a disease or medical condition, or the percentage of people who experience a given condition within a given time frame, e.g., 20% of people will get influenza in a year. 18. This measure of sickness is contrasted with the mortality rate of a condition, which is the proportion of people dying during a given time interval. Morbidity rates are used in actuarial professions, such as health insurance, life insurance, and long-term care insurance, to determine the premiums charged to customers. Morbidity rates help insurers predict the likelihood that an insured will contract or develop any number of specified diseases. Pathosis or pathology. Pathosis, plural pathoses, is synonymous with disease. The word pathology also has this sense, in which it is commonly used by physicians in the medical literature, although some editors prefer to reserve pathology to its other senses. Sometimes a slight connotative shade causes preference for pathology or pathosis, implying some as yet poorly analyzed pathophysiologic process, rather than disease, implying a specific disease entity as defined by diagnostic criteria being already met. This is hard to quantify denotatively, but it explains why cognitive synonymy is not invariable. Syndrome. A syndrome is the association of several signs and symptoms, or other characteristics that often occur together, regardless of whether the cause is known. Some syndromes, such as Down syndrome, are known to have only one cause, an extra chromosome at birth. Others, such as Parkinsonian syndrome, are known to have multiple possible causes. Acute coronary syndrome, for example, is not a single disease itself but is rather the manifestation of any of several diseases, including myocardial infarction secondary to coronary artery disease. In yet other syndromes, however, the cause is unknown. A familiar syndrome name often remains in use even after an underlying cause has been found or when there are a number of different possible primary causes. Examples of the first mentioned type are the Turner syndrome and DiGeorge syndrome, are still often called by the syndrome name, despite that they can also be viewed as disease entities and not solely as sets of signs and symptoms. Predecessors. Predecessors is a subclinical or prodromal vanguard of a disease. Prediabetes and prehypertension are common examples. The nosology or epistemology of predecessors is contentious, though, because there is seldom a bright line differentiating a legitimate concern for subclinical or premonitory status and the conflict of interest driven over medicalization, e.g., by pharmaceutical manufacturers, or demedicalization, e.g., by medical and disability insurers. Identifying legitimate predecessors can result in useful preventive measures, such as motivating the person to get a healthy amount of physical exercise. 19. But labeling a healthy person with an unfounded notion of predecessors can result in overtreatment, such as taking drugs that only help people with severe disease or paying for treatments with a poor benefit-cost ratio. One review proposed 3 criteria for predecessors. A high risk for progression to disease, making one far more likely to develop it than others are. For example, a precancer will almost certainly turn into cancer over time. Actionability for risk reduction. For example, removal of the precancerous tissue prevents it from turning into a potentially deadly cancer. Benefit that outweighs the harm of any interventions taken. Removing the precancerous tissue prevents cancer, and thus prevents a potential death from cancer. 20. Types by body system. Mental. Mental illness is a broad, generic label for a category of illnesses that may include affective or emotional instability, behavioral dysregulation, cognitive dysfunction or impairment. Specific illnesses known as mental illnesses include major depression, generalized anxiety disorders, schizophrenia, and attention deficit hyperactivity disorder, to name a few. Mental illness can be a biological, e.g., anatomical, chemical, or genetic or psychological, e.g., trauma or conflict origin. It can impair the affected person's ability to work or study and can harm interpersonal relationship. Organic. An organic disease is one caused by a physical or physiological change to some tissue or organ of the body. The term sometimes excludes infections. It is commonly used in contrast with mental disorders. It includes emotional and behavioral disorders if they are due to changes to the physical structures or functioning of the body, such as after a stroke or a traumatic brain injury, but not if they are due to psychosocial issues. Stages. In an infectious disease, the incubation period is the time between infection and the appearance of symptoms. The latency period is the time between infection and the ability of the disease to spread to another person, which may precede, follow, or be simultaneous with the appearance of symptoms. Some viruses also exhibit a dormant phase, called viral latency, in which the virus hides in the body in an inactive state. For example, varicella zoster virus causes chickenpox in the acute phase. After recovery from chickenpox, the virus may remain dormant in nerve cells for many years, and later cause herpes zoster shingles. Acute disease. An acute disease is a short-lived disease, like the common cold. Chronic disease. A chronic disease is one that lasts for a long time, usually at least 6 months. During that time, it may be constantly present, or it may go into remission and periodically relapse. A chronic disease may be stable, does not get any worse, or it may be progressive, gets worse over time. Some chronic diseases can be permanently cured. Most chronic diseases can be beneficially treated, even if they cannot be permanently cured. Clinical disease. One that has clinical consequences, in other words, the stage of the disease that produces the characteristic signs and symptoms of that disease. 21. AIDS is the clinical disease stage of HIV infection. Cure. A cure is the end of a medical condition or a treatment that is very likely to end it, while remission refers to the disappearance, possibly temporarily, of symptoms. Complete remission is the best possible outcome for incurable diseases. Flare-up. A flare-up can refer to either the recurrence of symptoms or an onset of more severe symptoms. 22. Progressive disease. Progressive disease is a disease whose typical natural course is the worsening of the disease until death, serious debility, or organ failure occurs. Slowly progressive diseases are also chronic diseases, many are also degenerative diseases. The opposite of progressive disease is stable disease or static disease, a medical condition that exists, but does not get better or worse. Refractory disease. A refractory disease is a disease that resists treatment, especially an individual case that resists treatment more than is normal for the specific disease in question. Subclinical disease. Also called silent disease, silent stage, or asymptomatic disease. This is a stage in some diseases before the symptoms are first noted. 23. Terminal phase. If a person will die soon from a disease, regardless of whether that disease typically causes death, then the stage between the earlier disease process and active dying is the terminal phase. Recovery. Recovery can refer to the repairing of physical processes, tissues, organs, etc. and the resumption of healthy functioning after damage-causing processes have been cured. Extent. This rash only affects one part of the body, so it is a localized disease. Localized disease. A localized disease is one that affects only one part of the body, such as athlete's foot or an eye infection. Disseminated disease. A disseminated disease has spread to other parts. With cancer, this is usually called metastatic disease. Systemic disease. A systemic disease is a disease that affects the entire body, such as influenza or high blood pressure. Classification. Diseases may be classified by cause, pathogenesis, mechanism by which the disease is caused, or by symptoms. Alternatively, diseases may be classified according to the organ system involved, though this is often complicated since many diseases affect more than one organ. A chief difficulty in nosology is that diseases often cannot be defined and classified clearly, especially when cause or pathogenesis are unknown. Thus diagnostic terms often only reflect a symptom or set of symptoms. Syndrome. Classical classification of human disease derives from the observational correlation between pathological analysis and clinical syndromes. Today it is preferred to classify them by their cause if it is known. 24. The most known and used classification of diseases is the World Health Organization's ICD. This is periodically updated. Currently, the last publication is the ICD-11. Causes. Diseases can be caused by any number of factors and may be acquired or congenital. Microorganisms, genetics, the environment or a combination of these can contribute to a disease state. 25. Only some diseases such as influenza are contagious and commonly believed infectious. The microorganisms that cause these diseases are known as pathogens and include varieties of bacteria, viruses, protozoa, and fungi. Infectious diseases can be transmitted, e.g. by hand-to-mouth contact with infectious material on surfaces, by bites of insects or other carriers of the disease, and from contaminated water or food, often via fecal contamination, etc. 26. Also, there are sexually transmitted diseases. In some cases, microorganisms that are not readily spread from person to person play a role, while other diseases can be prevented or ameliorated with appropriate nutrition or other lifestyle changes. Some diseases, such as most but not all forms of cancer, heart disease, and mental disorders, are non-infectious diseases. Many non-infectious diseases have a partly or completely genetic basis, see genetic disorder, and may thus be transmitted from one generation to another. Social determinants of health are the social conditions in which people live that determine their health. Illnesses are generally related to social, economic, political, and environmental circumstances. 27. Social determinants of health have been recognized by several health organizations such as the Public Health Agency of Canada and the World Health Organization to greatly influence collective and personal well-being. The World Health Organization's Social Determinants Council also recognizes social determinants of health in poverty. When the cause of a disease is poorly understood, societies tend to mythologize the disease or use it as a metaphor or symbol of whatever that culture considers evil. For example, until the bacterial cause of tuberculosis was discovered in 1882, experts variously ascribed the disease to heredity, a sedentary lifestyle, depressed mood, and overindulgence in sex, rich food, or alcohol, all of which were social ills at the time. 28. When a disease is caused by a pathogenic organism, e.g., when malaria is caused by Plasmodium, one should not confuse the pathogen, the cause of the disease, with disease itself. For example, West Nile virus, the pathogen, causes West Nile fever, the disease. The misuse of basic definitions in epidemiology is frequent in scientific publications. 29. Types of causes. Regular physical activity, such as riding a bicycle or walking, reduces the risk of lifestyle diseases. Airborne. An airborne disease is any disease that is caused by pathogens and transmitted through the air. 30. Foodborne. Foodborne illness or food poisoning is any illness resulting from the consumption of food contaminated with pathogenic bacteria, toxins, viruses, prions or parasites. 31. Infectious. Infectious diseases, also known as transmissible diseases or communicable diseases, comprise clinically evident illness, i.e., characteristic medical signs or symptoms of disease resulting from the infection, presence and growth of pathogenic biological agents in an individual host organism. 32. Included in this category are contagious diseases, an infection, such as influenza or the common cold, that commonly spreads from one person to another, and communicable diseases, a disease that can spread from one person to another, but does not necessarily spread through everyday contact. Lifestyle. A lifestyle disease is any disease that appears to increase in frequency as countries become more industrialized and people live longer, especially if the risk factors include behavioral choices like a sedentary lifestyle or a diet high in unhealthful foods such as refined carbohydrates, trans fats, or alcoholic beverages. 33. Non-communicable. A non-communicable disease is a medical condition or disease that is non-transmissible. 34. Non-communicable diseases cannot be spread directly from one person to another. Heart disease and cancer are examples of non-communicable diseases in humans. 35. Prevention. Many diseases and disorders can be prevented through a variety of means. These include sanitation, proper nutrition, adequate exercise, vaccinations and other self-care and public health measures, such as obligatory face mask mandates. Treatments. Medical therapies are treatments or efforts to cure or improve a disease or other health problems. In the medical field, therapy is synonymous with the word treatment. Among psychologists, the term may refer specifically to psychotherapy or talk therapy. Common treatments include medications, surgery, medical devices, and self-care. Treatments may be provided by an organized health care system, or informally, by the patient or family members. Preventive healthcare is a way to avoid an injury, sickness, or disease in the first place. A treatment or cure is applied after a medical problem has already started. A treatment attempts to improve or remove a problem, but treatments may not produce permanent cures, especially in chronic diseases. Cures are a subset of treatments that reverse diseases completely or end medical problems permanently. Many diseases that cannot be completely cured are still treatable. Pain management, also called pain medicine, is that branch of medicine employing an interdisciplinary approach to the relief of pain and improvement in the quality of life of those living with pain. 36. Treatment for medical emergencies must be provided promptly, often through an emergency department or, in less critical situations, through an urgent care facility. Epidemiology. Epidemiology is the study of the factors that cause or encourage diseases. Some diseases are more common in certain geographic areas, among people with certain genetic or socioeconomic characteristics, or at different times of the year. Epidemiology is considered a cornerstone methodology of public health research and is highly regarded in evidence-based medicine for identifying risk factors for diseases. In the study of communicable and non-communicable diseases, the work of epidemiologists ranges from outbreak investigation to study design, data collection, and analysis, including the development of statistical models to test hypotheses and the documentation of results for submission to peer-reviewed journals. Epidemiologists also study the interaction of diseases in a population, a condition known as a syndemic. Epidemiologists rely on a number of other scientific disciplines such as biology to better understand disease processes, biostatistics, the current raw information available, geographic information science to store data and map disease patterns, and social science disciplines to better understand proximate and distal risk factors. Epidemiology can help identify causes as well as guide prevention efforts. In studying diseases, epidemiology faces the challenge of defining them. Especially for poorly understood diseases, different groups might use significantly different definitions. Without an agreed-on definition, different researchers may report different numbers of cases and characteristics of the disease. 37. Some morbidity databases are compiled with data supplied by states and territories' health authorities, at national levels 38, 39, or larger scale, such as European Hospital Morbidity Database, HMDB, 40, which may contain hospital discharge data by detailed diagnosis, age and sex. The European HMDB data was submitted by European countries to the World Health Organization Regional Office for Europe.